Hi. Before the episode, we'd just like to thank everyone who would like to support Future Eminent. We couldn't do this without you, and we hope you keep supporting us. Please visit our Patreon or Ko-Fi page linked below when you feel like we've earned it. Now, on to the show. Biohacking. It involves doing most of the things that a giant lab would do, except without any of the budget. David Ishee has been working on a method to affordably alter the genetics of pure-bred dogs so that the diseases that come with the lack of genetic diversity can be removed. Listen in on our conversation about just that and the future of genetics and biohacking here today on Future Eminent. How are you doing today?
I'm doing pretty good. Tired. A long night in the lab last night.
You've gotten a lot of attention in the news. Specifically, I also want to talk about Unnatural Selection, that show on Netflix. Has the response been mostly positive or mostly negative?
Overwhelmingly, mostly positive.
How about from Unnatural Selection?
Yeah, it depends on what group we're talking about. Like the average everyday person, it's been very, very positive. There's a few academics that didn't like it.
How have traditional dog breeders reacted to the idea of biohacking their purebred dogs?
It varies. Some dog breeders don't like the idea of sort of altering a dog's DNA in any way, which includes crossbreeding or anything. It's kind of built around the idea that they're preserving some historical artifact and that any changes are necessarily destructive to that preservation. But I think most dog breeders, when you're talking about health, understand that like that's not the thing that we want to preserve, right? We don't want to preserve genetic diseases. In that aspect, people have been pretty positive. And then the more adventurous dog breeders who are more focused on the future and less on the past are excited about the possibilities of what we can do with a broader selection of genes to work with.
Are they educated on the fact that the pure breeding is still resulting in these genetic disorders being carried on through the generations? Are they at least willing to admit that?
That's a harder one to swallow. Like there's a growing number of breeders who understand that highly isolated gene pools is a bad thing. That genetic diversity is a good thing. But I'd say the majority of the sort of professional breeders are still pretty dyed in the wool with the purebred camp. The only reason to breed is to meet the show standard and all this sort of stuff. But working breeders and the designer dog community and a bunch of other people who are more focused on function and health are much more open to the idea of destigmatized non-pure breeding, if you want to think of it that way. In agriculture a long time ago, if someone crossbreeds between two different kinds of pig, nobody cares. It's all about what you want out of the pig. If the pig's a meat animal, then whatever produces the most meat, whether it's a purebred pig or not, is kind of irrelevant. There actually are still livestock shows. There's some amount of pure breeding involved because there's like show pigs, but for the most part, goats and cows, pigs and things like that are bred for function, not for show. And so I think that's part of what drives the cultural difference there.
So one of my favorite quotes that I heard, your description of genetic engineering as this ocean, and that to make any headway you need to have a destination. So your destination seems to be removing genetic disorders from Dalmatians, was it?
That's just sort of the local destination, but yeah, it's one stop. There's a genetic disease that's in basically all Dalmatian. But they're sort of like the first test case. The real goal is to get rid of all of them in all the breeds. You know, we have the tools and we have the technology. It's just a huge engineering problem and it kind of coupled to a social and cultural problem. How do you solve all of that at once? So that's the bigger goal. Dalmatians is just one part of that. The main thing is, say you cure Dalmatians in a couple of years of hyperuricemia. I think that creates a sort of a precedent that you can then start arguing with basically all the other breeds and saying, hey, what about this disease? What about that disease? Let's see how we can apply this and then get people working together. Because that's what it's really going to take. It's not about me tinkering in my lab, trying to cure one disease at a time. There's too many. I'll never get it all. The goal is to create a process, groups and infrastructure where people can come together and work together on these problems and use advanced tools and solve them collectively, because there's way more work than I can ever do.
Another thing I wanted to touch on was bioluminescence. I know that's not your goal. It's just the way you put it was to this procedure that you're trying to develop is working. What is the difference? You've also mentioned that there's a difference between, different animals in terms of genetic engineering. So could this be used for other species? A good example is that Aqua Advantage that took 20 years to get FDA approval, but I assume they didn't use bioluminescence to showcase that hey this works and see
No.
there's nothing. Yeah, exactly. So maybe bioluminescence is like a good idea to showcase to maybe people that don't understand genetic engineering as well that there's nothing really too harmful about it, just that hey it worked here and now X result, now these people don't have this genetic disorder. Well, maybe not use bioluminescence on people, but basically
Not impossible, but yeah.
so
I know quite a few people who at least like a little bit of bioluminescence, like a few spots here and there.
hey, I'm not saying I'm necessarily against that, but so basically, how would you take this procedure and adapt it to other animals in the animal kingdom?
If you're looking at the particular technique that I'm developing, it's very sort of dog centered, but it doesn't necessarily have to be. So basically, you just have to repeat what I did. Now, if you wanted to use it on something like pigs, for example, that would be relatively straightforward because it's already been done on pigs. That's sort of where I'm taking it from. My whole project is translating this technique that was developed to modify the sperm instead of the egg in pigs and apply it to dogs. Dog sperm and pig sperm are pretty different, so it takes a while. But if you wanted to, say, apply it to cats or something else, then you just have to figure out what changes need to be made to the protocol to make it work in cats. And do that, then you have a method that works in cats, and then you have a method you could use in horses or whatever. But the whole point of using something like sperm-mediated gene transfer is you don't have to put the animal to sleep. You don't have to extract eggs. You don't have to do in vitro fertilization. You don't have to have embryos in a freezer somewhere. You don't have to clone anything. You just collect sperm, modify the sperm, and artificially inseminate. So it's much more approachable as a breeder. And so I think that's the reason I focus so heavily on that particular protocol. Like right now, sort of the standard method, like say you wanted to make a genetically modified bull, you would collect some skin cells from the bull, grow them in a petri dish, genetically modify those skin cells, test them, make sure that they have the modifications that you want, collect an egg cell from a bull, from a bull, collect an egg cell from a cow, sort of suck out the nucleus, all the DNA, take the DNA out of that skin cell and put that nucleus into the egg cell. So that's somatic cell nuclear transfer. Then give that egg cell with the modified nucleus, modified genome, a little electrical zap, and then it starts to become an embryo. And then once it grows into a large enough embryo, you implant that in a cow and she carries it to term. And that's genetically modified clone at that point. But that's much more involved and really outside the reach of breeders for quite some time and much more expensive. So the standard protocol, if you just wanted to do something big, like a project on the scale of Aqua Advantage, where you're going to be putting something out in the market where it's like, oh, we make genetically modified cows and they produce more milk or produce better meat or something, that would be something that could pay for the sort of typical techniques. Regulatory obstacles are still going to be a big issue. In terms of trying to cure a bunch of diseases that don't have significant economic drive, like hip dysplasia in dogs or something like that, really don't have the many dogs that would have to be cloned and genetically modified. You'd never be able to pay for it without some sort of like massive government grant or some corporation giving huge amounts of money towards it, and that's just not going to happen.
Gotcha. So it's two different worlds, the aqua salmon versus what you're doing.
Exactly. And so like the Aqua Advantage salmon, like it was pretty obvious that their thing worked because first of all, the fish are triploid, I'm pretty sure. They may be tetraploid. Basically, they have more copies of the genome, so you can look at their cells under a microscope and tell that they're different. And then they're also way bigger in a shorter amount of time. You can tell because they outgrow all their cousins on less food. So that leaves a pretty distinct marker, but just curing a disease doesn't really leave a distinct marker. So if you show up with a dog and say, hey, look, I've got a dog who's not sick, it doesn't really look like anything. So you can't just show up at a breeder and say, hey, my dog isn't sick. Don't you wish your dog to be like that? While they would say yes, you say it only took genetic engineering. And I think they really won't quite grasp possibilities because like there's all these different ways with existing tools to remove genetic diseases, but they all sort of destroy the genetic diversity of the population and take forever. If you can say, here's something that you never expected to see, a dog that's part jellyfish and glows, then I think you can really get them to see the potential and think outside the box a little bit. And I think that's going to be a big, important next step for dog breeding in general.
Most recently, you mentioned having issues with a new CRISPR template. What makes it different from when CRISPR initially came out in 2013? And what are those hangups, more specifically? CRISPR technology is a simple yet powerful tool for editing genomes. It allows researchers to easily alter DNA sequences and modify gene function. Its many potential applications include correcting genetic defects, treating and preventing the spread of diseases, and improving crops.
The issues I was having weren't specifically with CRISPR. So that has to do with a different project of mine, trying to make DIY genetic engineering more accessible. And again, it's about building infrastructure and reducing the sort of costs and barriers to entry. So one of the things I needed to do in that time was to modify a strain of bacteria that are used to produce the gene therapy to make them better at doing that job. So I was using CRISPR to modify their genome. But the issues I was having were more about the back end molecular technologies like PCR. And because I was trying to get the template, which is the thing that's going to be added to their genome, I was trying to get it ready and making sure that it's right is there. But it's not generated by CRISPR, it's generated by a different technique. You got to do all the lab work beforehand to have all the pieces. But what I was doing was pretty standard CRISPR as it was. Now, there's a lot of different versions of CRISPR that have come up since original release of CRISPR-Cas9. That's the most interesting part of it, I think, because there's things like prime editing, base editing, and all these different versions, because there's things where you can just use a modified Cas9 to turn genes on and off, or edit methylation, or do all these programmable modifications by sort of hijacking Cas9's ability to seek out a particular sequence, bind, and then do something. You can kind of use it as think of it like a truck where you just attach a trailer to it and it carries it to the right spot. And then the other enzyme will then do its job in that place. There's a lot of different versions, and that is just the beginning, because there's going to be more versions where they change its binding affinity, they change ham sites, they find more Cas proteins that have different abilities and that are smaller and more efficient and will fit into viruses and things like that. But generally, the CRISPR that I'm using is pretty standard CRISPR. Cut the genome, and then I'm doing a knock-in. So you provide a template so that when the cell repairs itself, it uses my template as a guide to do that, and it ends up with the modifications that I made to the template get integrated during the repair process into its own genome. I know that's exciting.
I mean, if it's not broke, why fix it, right?
The thing that I'm trying to do isn't particularly advanced. I mean, that's not really new. Now, I probably will use a mutated Cas9 when I actually do the CRISPR for the Dalmatians, because there's a high fidelity Cas9. One of the things that got a lot of attention and is maybe a little overblown, but still something to be concerned over, is off-target effects. So Cas9 is sort of a programmable scissors. You tell it where to cut, and it goes in there, finds that spot, and cuts it. But it's not perfect. It will cut things that are very similar, or if you just have a lot of it, or leave it in there too long, or if there happens to be another spot with the same sequence, it can cut the wrong place. There was a paper done pretty shortly after the original Cas9 sort of started gaining ground, where they did various mutations and checked for fidelity. So I checked the amount of off target. And so there's a version of it that I have that's Cas9, but it's mutated in such a way that its off target effects are less than the sort of base mutation rate. You can do CRISPR, and the mutation rate doesn't go up from what it would if you just had two cells divide, right? So, I mean, a cell divides, there's a base mutation rate. It might mutate X probability, and doing Cas9 with that version doesn't raise it. So there's basically no off target, because you don't want to genetically modify them to repair a broken gene just to have the tools that you're using go somewhere else and break some other gene.
Now, jumping over a little bit, I heard that you and a guy called Joshua Zahner, Josiah, wanted to make a dog gene sequencing company. How is that going?
Yes. Slowly.
Josiah Zahner is a biohacker and scientist best known for his self-experimentation and his work making hands-on genetic engineering accessible to a lay audience, including CRISPR.
So, I have 22 genomes, right? They're all my dogs. The idea there is to provide breeders with tools to help them do a better job. Imagine you could, if you were a dog breeder, one of the things I get called about is I get called to sort of model people's breedings. So let's say I've got, these two dogs, I want to breed them together. Can you tell me what to expect in my litter? They'll tell me about the dogs. They'll tell me the dogs' colors and their parents' colors and so on. And I'll be able to say, okay, well, half of them will be this, a quarter of them will be that, and so on. The thing there's pretty simple, because knowing the relationships between the genes and which are dominant, which are recessive, and which parent has what. Usually you can tell there's some things obviously both carry for something recessive that hasn't popped up in generations, could sort of pop up in some surprise you. But if you sequence the genes of the dog, then you not only know what the dog has that you can see, but also the things that the dog carries. And if two dogs carry for, say, a genetic disease, they may be both perfectly healthy, but both carriers. You breed them together, and now you start getting dogs who are afflicted. So it would be nice if dog breeders could sequence their dogs cheaply and then simulate their breedings. So they say, okay, well, they pull up their thing, they've got 5 different dogs in their account, and they say, okay, what if I breed this pair? And it says, okay, well, this is what you can expect for puppies. And also, here's a red flag, you might have this issue. Maybe this isn't the best breeding. Then they can simulate other breeding or even share that with other breeders so they can simulate breeding. So that way, it helps the breeders see into the spaces that are normally invisible because you can't see what the dog carries, the recessive things. It helps dog breeders to predict things that are very difficult to understand. So like you can make a normal dog breeder with a little infrastructure be able to breed as well as a world-class dog breeder. And so giving them the tools to help them do a better job just leads to fewer sick dogs being produced, better outcomes for the breeders, better outcomes for the dog, and better outcomes for the people that get dogs from them. So everybody wins, and you can create a database of dog genomes, thousands of them. And when you get enough of them, you can start doing machine learning on and start trying to sort of figure out where all the sort of knobs and levers things are. And so if you make this change, the dog changes like this. If you make that change, the dog changes like that. And so there's a lot of things we know and there's a lot of things we don't know. Having that information in a nice consolidated place would give you the ability to start finding those things out. And if you get that, then you're in a position where you can say, okay, this is the dog I'd like to have. You can have the machine spit out the genes that you're going to need to do that. And if you can do that, then you're in a position to really make the best dogs that have ever been. And I think that's really powerful.
Hey, they're healthy at the same time, so it's perfect.
Exactly. They'd be healthy and they'd be able to do their jobs better. So if you've got like a search and rescue dog, don't you want, if you're buried under an avalanche, the best search and rescue dog ever born to be the one who's coming to save you? If you're a bomb sniffing dog, don't you want the best bomb sniffing dog ever to be the one patrolling the area you're in? So like you've got a working dog, you want them to be the very best at their job and have a good quality of life, a nice, long, healthy life, enjoy their jobs. All that's possible. It's just difficult breeding. And I think it really comes down to the tools, giving people the tools to do a better job.
Has the recent COVID-19 pandemic changed anything in your conversations with the FDA? Because I did read an article how back in 2017 you guys had a back and forth that could have gone better. The FDA had told him to not sell his Dalmatians and to keep them on his property. Their policy with all genetically engineered animals is to keep them under quarantine.
Yeah, I mean, the FDA still looks at dogs as drugs, which is silly. But I think, I don't know, maybe they've got more on their plate. But it really hasn't changed anything. They're busier, probably, because there's like 70 different coronavirus vaccines being tested right now. That's probably got their main focus. It really hasn't changed anything on the sort of genetically modified animals front. I do think it leaves discussion about genetically modified people, though, because out of China, there were 2 babies born modified with CRISPR, genetically engineered to be resistant to HIV. So these 3 babies were genetically engineered to be born resistant to a global pandemic. It's a virus that's killed, how many people has HIV killed in its time? And so I just wonder, at that time, everybody was pretty upset about the idea of genetically modified humans, even if the idea was to make them resistant to a virus. And I just wonder if people have the same reservation as they did a year ago. I feel like people are a little more open to the discussion of making humans who are virus resistant.
My last main question that I wanted to ask you is, are you aware that you're sort of democratizing genetic engineering? And a lot of people are scared about a society in the future where it's going to be the rich that have access to genetic tube babies, or whatever the term you want to use. First,
Right.
what are the pros and cons of that happening versus, let's say you come up with a procedure that eventually people can use on human beings. And then eventually, you're a nice guy. You wouldn't go against the well wishes and well beings of the general public. But somebody out there that's a bit more malicious would start. In China, for example, there aren't exactly laws against it. Somewhere in the world, somebody is going to have these tools and will easily alter human beings. They could either do it for a genetically brain dead but very capable army that's only intelligent and using weaponry. I mean, a bit of an out there idea. Or I want to make somebody that's resistant to all disease and will live 200 years and be healthy their whole life, but they don't want to wait for government approval and they go ahead and do it anyway. I'm just wondering, what do you think is more likely? What do you think is realistic? And what would the pros and cons that people don't typically think of when thinking of those scenarios?
So this is something I think about a lot, and something where people don't really know where we are in that timeline. Everybody was shocked when CRISPR babies were born, but they were born basically exactly when I thought they would be. Because if you were looking at the papers coming out of China at the time, they were doing everything leading up to it, right? So it's like, okay, we CRISPR modified an embryo or an egg and then an embryo and grew it for 14 days and so on and so on. It's like the only thing left to do is implant one and bring it to term. And there's a lot of things that a lot of people just aren't aware of unless you're really following it closely. So it feels like things that are 20 years away just keep happening. But I think the scenario of a social disparity is a very real one, because obviously we already have several of those going right now. And one of those is in medicine. It's one of the things that they talked about in the documentary is the expense of things like gene therapy and accessibility. And why does it cost $1 million for an injection that cost about $5,000 to make? Like, what are the sort of systemic and cultural factors that lead into those kind of situations? And why do we accept them? And I think the ability for someone to say, okay, I have access to this technology, I can do amazing things with it. I'm either going to use it for myself, even if it's not legal, I'll just go where it is legal, is not only likely, but it's happening now. Like, I know people who are leave the country, go get stuff done. I know people trying to set up places for genetically modified babies and things like that for rich people. I know there are people who will try to regulate it, and I think that will have sort of a negative impact on trying to reduce that disparity, because when you say, okay, this is just not allowed, then that just means the people who have enough resources to break the rules, either by going to their own private, Epstein Island and do whatever what they want, far away from regulations and law, or someone will just go to some country that doesn't have any laws against it. Some countries will use that as medical tourism kind of thing. Like right now, there are surgeries that people go and do. It really is like gene therapy and genetically engineered babies is a little bit more like surgery than drugs, for example. Not that they've been able to really control drugs, but they certainly can't control surgery. You go to Mexico, get your lap band surgery and come back, they don't kick you out for that. And so it'd be the same thing if you went to Mexico, got your genetic modifications and came back. That stuff's happening now. There's a reason the World Anti-Doping Agency is testing all the Olympians for what they call gene doping to make sure they haven't genetically modified themselves to be better athletes. As far as soldiers and state things, that's probably the scariest, because that's also happening. Like right now, there's a company, Rejuvenate Bio, who's doing some stuff with dogs right now, but like they have DARPA funding. In their grant award, you can read that their first things they're working on is multi-purpose canines, so scent and bite work dogs, dog soldiers basically, to make them better soldiers. And they have 3 phases. So they did it with mice, doing with dogs, and their next phase they already have like it's over $1 million, which isn't a lot of money, but the fact that DARPA is paying a company to do tests on US soldiers with gene therapy to make them better soldiers, like we are making super soldiers. It's not in large numbers and it's all just DARPA stuff right now. If that's what the US is doing openly, what's happening around the world secretly, who knows? But that's happening. And just this idea of faster, smarter, stronger, tougher soldiers is only a tiny piece of what's possible. So I do think we'll see a situation where there's military applications for this kind of technology where rich people can go get whatever they want. There has to be some sort of relief valve for that, because it creates a lot of disparity in power. The average person, maybe you can't afford to go get the $1 million gene therapy in Taiwan. And maybe you're in a position where you desperately need some gene therapy, or you just want your children to have sort of a competitive chance in the future. I think that's where it's important to either have looser regulations where people have a choice, or you have to have some way to do it yourself. That way, there's at least some option to sort of get into that, because otherwise you create a situation, like, for example, with virus resistance. There is a way to make a human completely resistant to all viruses. In fact, it's something that George Church's lab's been working on. They have made E. coli, which admittedly is a long way from a human, that are resistant to 80% of all viruses that affect E. coli. And the genetic changes necessary to do that would be pretty drastic. But if you basically recode the codon, your cells no longer speak the same language as viruses. You could make a human who was a human, but completely resistant to all viruses, whether they're in humans now or they're in another mammal and they'll jump to humans in the future. And they would just be virus immune forever. And all their children would be and everything. The problem is they would not be able to reproduce with normal humans. you would basically have to make a new species of human. Even if the only change you made was to make them immune to all viruses, I don't know how we would respond to that socially. I don't know how we would respond to that politically. But if you were in a position where all the wealthy and all the powerful were basically a different species from us and had this super disease resistance, I don't know if that would lead to some serious civil unrest or not, but I think in order to avoid that future, which I think is probably more likely than not, I think there has to be like a relief valve. There has to be some way for that pressure to go off. And I think the only way, some people are like, well, let's just shut it down and never do that. But that's terrible. Like, we would prefer that all humans be immune to viruses. Like, why should people die of viruses? But the issue is, how do we create access? How do we create a path for the average person to be able to get these things? And that's, even if you never get it through a black market, I think having like a DIY or black market or something like that forces companies that are doing it like through the FDA, through sort of regulatory pathways, to reduce like what they charge. Because you know that if you charge $1 million, people are just going to do it themselves. So basically, I think just knowing that there's another route will cause companies to reduce their prices, because if you make your prices unattainable, then people will go another way. If there is no other way, then you kind of have a monopoly. It would be like weed stores, right? Like, it's legal in Colorado, but you could still sell it illegally, right? By just not having a business and not paying your taxes. But if all the weed stores in Colorado sold weed for like $800 a gram or something like that, people still wouldn't go there. They'd just go to the same guy that they were getting it from before, right? So if you have another option like that, even if it's not the preferred option, I think it's still competition. And those companies still know it's there. And that still creates an incentive for them to reduce the price.
So we've come to the end of our list of questions. If there's anything else you'd like to talk about, or anything else you'd like to tell our listeners, could be anything at all. It could even be what you had for breakfast. I really don't care. It's the floor is yours.
See, I like interviews better because I'm better at answering questions and just coming up with things to say. There is another project that me and Josiah are working that I can't get into yet, but I think it'll be interesting. And I think in the next, should be a few weeks, we'll be able to start talking about that. So that ought to be exciting.
That was David Ishee, correct?
Ishee.
Sorry, David Ishee.
It's okay. About half the people say it that way, but so I'm used to it.
Thanks for coming on to the podcast.
Yeah, man.
Yeah, have a good one.
It was fun. You too.
I don't do this alone, and I wouldn't want to. As the host of Future Imminent, it is my pleasure to thank Nick Gertson for producing, Lee Collings for his help in writing our script, and the Futurist Foundation for supporting this podcast. Be sure to check out the description for the links to our website, our Discord channel, and our sponsors.